My name is George Weiner. I'm from the University of Iowa, and it's indeed my pleasure to moderate the session on clinical trial design for PD-1, PD-L1 combination therapies, something obviously we've already talked about to varying degrees this morning. So we have 4 presentations this afternoon, followed by a panel discussion. So Adil Daud will start out by talking about immunotherapy response and biomarkers. Ahmad Tarhini will talk about master protocols for immunotherapy combinations. And then we have our mini presidential symposium with Lisa Butterfield, who's president of SITC, talking about strategies for incorporating biomarkers in clinical trials, and Liz Jaffe, who's president of AACR, talking about appropriate endpoints for these studies. So a couple things I really hope we're able to cover this afternoon. It is talking not only about how we design trials to see if new regimens are tolerable with acceptable toxicities, if they're effective, what the clinical biomarkers are, but also talk about novel approaches to modular design, talking about how to design studies that are as economical as possible, and also talk a fair amount about how we can design studies so that we learn something even if the primary clinical endpoints are not met. I mean, the number of variables is huge for designing these trials: patient selection, drug dose, drug sequencing, correlatives to do, endpoints, et cetera. So I'm not going to spend any more time, and I'm really looking forward to the presentations and then the discussion. So Dr. Daud from UCSF will start out talking about immunotherapy response and biomarkers.
Thank you, Dr. Weiner. Thanks, everybody, for attending, and thank you, Dr. Clafe and Dancy. What I was going to do is to give just a flavor of where we are in terms of biomarkers and response to immunotherapy. So I think, as all of you in this room know, that PD-1 has been an incredible success, but it's not like every single person responds to it. And it's not like every single response is long-lasting. And this is data from the Keynote-001 study kind of illustrating that. And although we are talking about biomarkers today, you actually don't need biomarkers to a great extent to tell whether somebody is going to respond or not. When somebody walks into your clinic, you can already tell whether they're going to respond or not. And we published this paper last year, and just shows the influence that liver metastases and male gender have, and things like ECOG, performance status. So a lot of times I can tell that if a 75-year-old guy with lung metastases from melanoma walks into my clinic, he's going to respond. And if a 25-year-old female walks in with bulky liver metastases and elevated LDH, she will not respond. You don't need any type of microscopic examination of the tumor to tell you that. And our gender analysis has been reproduced several times. This is a paper by Goldhirsch in showing this is true across multiple trials and disease types. And then we also showed that patients who have liver metastases don't respond, and that doesn't have much to do with the biomarker influence on the disease. But what we showed is that if you have liver mets only, and you could see that in the bottom line, if you have liver mets, your PFS is 2.96 months. If you have just lung mets, it's 23 months. So that's a 10-fold difference, and that's nothing to do with microscopic examination of the tumor. And what we've shown is that if you look at partially exhausted, or what we've defined as CTLA-4 and PD-1 dual positive CD8 T cells, you see a lot more of those in patients who don't have liver metastases. So we think that could be a partial explanation. So I know you guys have just seen this, but we know that, and this is for pembrolizumab, we know that pembrolizumab is not active equally throughout all tumor types. And we also know that PD-L1 is a good biomarker, and this is just data in melanoma. You could see that both for response and for overall survival and progression-free survival, all three of those vary a lot depending on your PD-L1 status. So what can we learn further? So I think at the outset it should be clear that we are talking about multiple different types of immunotherapy. PD-1 immunotherapy, obviously, you're talking peripheral tolerance, while CTLA-4 antibodies, you're talking about the priming phase, right? And if you look at PD-L1 expression, and this is from IPI-NIVO trial, you could see that based on the PD-L1 expression, for patients who are low-level PD-L1 expressors, you could see that there is a significant difference between IPI-NIVO and between NIVO. That's the orange and the blue lines. While for those who are high-level PD-L1 expressors, there isn't a lot of difference. So looking at further signatures of response, I think the one very significant piece of data has been this interferon gamma signature, which has already been remarked upon. And while the interferon gamma signature won't segregate patients, perfectly, it does segregate patients to some extent. And to me, what has been very interesting about that signature is that it's very similar. If you look at some of the CXCLs, like CXCL 9, 10, 11, and CCL5, it's actually very reminiscent of the signature that's been published, and those of us in the vaccine development field know already from 2011 as a marker for tertiary lymphoid structures within the tumor microenvironment. So these are not lymph nodes; these are just lymphoid aggregates that are found in the tumor microenvironment. And this is the paper by Domenico Coppola and Jim Muley, just showing the presence of these lymphoid aggregates. They're basically collections of B cells and T cells, and interestingly, very similar to the gamma interferon signature. So one other point that needs to be made, and I think somebody has made this already today, is that the order of administration of checkpoint inhibitors makes a huge difference to the ultimate response rate and outcome. And this is actually overall survival. This is a trial that Jeff Weber and Steve Hody did along with their colleagues, where patients were either given IPI followed by NIVO or NIVO followed by IPI. That's the only difference. So same agent, and cohort A is basically NIVO followed by IPI. Cohort B is IPI followed by NIVO. That's the only difference. And you could see the significant difference in overall survival. What was interesting is, and Steve Hody showed this a couple of years ago, and Scott Rodig has shown this data as well, that if you look at the biomarkers that predict responses to the NIVO first cohort of that trial do not hold true for the IPI first cohort of that trial, which is very interesting to me. And many of the biomarkers that they described, similar as the interferon gamma profile, like CXCLs 9, 10, 11, and 13, and some of the TNF family members, IDO1. So, but the only difference here is that the order is different, and this just illustrates that kind of in one graph here, that if you look at NIVO followed by IPI, the class 1 expression, T cell infiltration, PD-1 expression, PD-L1, none of those matter. And what's interesting is if you look at the tumor mutation burden, that's the last thing on the last row. Tumor mutation burden is important for NIVO-IPI. It is also important for IPI followed by NIVO, but in the opposite order. So in other words, the tumors that had low mutation burden seemed to respond better when IPI was given first followed by NIVO, but not when NIVO was given first followed by IPI. So kind of a thought-provoking thing, at least to me, that how could TMB be important, but in the inverse, in the obverse orientation, right? So we've taken a different tack. We know from previous data that T cells in the setting of cancer often don't clear the infection, and antigen persists, and they basically undergo this exhaustion. And so we figured that if you could extract T cells from tumor and determine if they had some of these exhaustion markers, that could be a marker for PD-1 response. And so that's the workflow that my colleague Mike Rosenblum followed in his lab. Essentially, we take a core or a punch biopsy, we digest it, and then we use flow cytometry. And we showed this a couple of years ago now. But basically, if you look at at CD8 T cells that are dual positive for PD-1 and CTLA-4, they appear to be highly enriched in responders. And if you look at the cells that are low expressors of PD-1 and CTLA-4, they are highly enriched in non-responders. And you could just see that in the first two bars there, and just show how significantly different they are. Let's see if I can show that. So that's basically, these are the dual positives, and then these are the dual negatives. So you could just see this obverse relationship. And if you qualify that or quantify that, you could see that a lot of responders look like this, and a lot of non-responders look like this. They have a lot of these CD8 cells that don't have these dual markers on, while a lot of responders look like this. And you can see in the discovery cohort and validation cohort, this holds pretty well. And essentially, this is the kind of graph that you get. If you look at patients who are below this 20% level, so that's another way of saying less than a fifth of all T cells in the tumor microenvironment have both of these exhaustion markers. None of these patients responded. If you look at patients over 30%, so more than a third or more have these exhaustion markers, everybody responded. And between 20 and 30%, there's some play in our system. And so, you could have a response, you could have a non-response, and you could look at some of the other characteristics too, like whether they have got LDH or and not surprisingly, a lot of the deaths are concentrated in the right-hand side. So we've gone on since then and extracted T cells from patients and bulk sequenced them. And basically, what you see is that these T cells have a very distinctive genetic and expression profile compared to these cells. And they express a lot of the markers that you would expect that they express. But some things are somewhat surprising. Like, for instance, there's a lot of proliferation going on in the quote-unquote exhausted T cells, because to some extent, I think the word exhaustion is misleading. I think these are just chronically activated T cells, and I think they're part of the proliferating cells in that pool. And this just shows the different expression profiles of these cells. So lately, we've gone on and we've collaborated with, and this is work from Mike Rosenblum and from folks at the Broad Institute. And this is a single-cell RNA-seq profile from this one patient. She's a patient who progressed on PD-1 and had bulky disease left. So that's why we were able to get tumor out and run this. And this is from the CD45 positive channel. And what you could see is that this is all the CD8 cells. Those are Tregs. Those are CD4s. And so you could see that even in the CD8 pool, there's a lot of interesting variations. So if you look at PD-1 expression, it seems to be concentrated in the lower half of this pool. But if you look at the TCR expansion, so these are just the two most common clones that are expanded. And we think these TCRs represent tumor-specific TCRs because of their expansion, just simply because they are expanded so much. And you could see that if you look at this TCR, doesn't seem to have a lot of PD-1 expression. If you look at this TCR, maybe there is some overlap. And you could see that with some of the other, like TIGIT, seems to be expressed in Tregs and also in the exhausted T cells. Or if you look at TIM3, that seems to be pretty much concentrated in the lower half of that T cell pool. So I think the point I'm trying to make here is that when you get below the bulk data and look at individual clones, there are some clones with TCRs that are expanded that don't express PD-1 and could be reactivated with different checkpoints, even in patients who have progressed, in this one case, progressed on PD-1. And this just shows some other markers that we have looked at too. This is CD39, and you can see that CD39 may be a better marker, and others have noticed this too, and may be coinciding to some extent with this particular pool of TCRs. So what else can we learn from flow cytometry in tumors? This is a study that Max Krummel, one of my colleagues at UCSF, did. And basically, what we looked at here is is looking at these cross-presenting dendritic cells, or what we now call CDC1s, or BDCA3 expressing dendritic cells. And you can see that if you look at the responders versus non-responders, responders seem to have a lot more of these dendritic cells compared to non-responders. And that's just another way of looking at it. Total CD45 positive cells don't seem like they're a lot different between responders and non-responders. But these BDCA3 positive cells are a lot more concentrated in responders. And so this is the model that we've evolved, which is that if you have a lot of these cross-presenting dendritic cells, it seems like that goes along with having more of these exhausted TILs. And if you have more of these MDSCs, or non-cross-presenting T cells, you don't have as many TILs, or you have anergic TILs. And so this is a study we just published recently. And again, this is from Max Krummel's group, where basically what he's shown is that the presence of NK cells in many instances correlates with having a lot of these cross-presenting dendritic cells. And so he looked at head and neck cancer and melanoma. So this is melanoma and head and neck cancer. And basically showed that the presence of FLT ligand correlates with this NK cell signature. And the presence of NK cells correlates pretty well with having these cross-presenting DCs. And he's shown that in a variety of mice models, either by knocking out interleukin-2 receptor, or in a RAG knockout, where you basically preserve the presence of these cross-presenting DCs, but basically abrogated in an NK cell antibody-treated B16 tumor. And so this is the tumor. So this is the model we are working with right now, is that somehow these tumors attract the presence of NK cells, which in turn attract stimulatory DCs, and that triggers the presence of PE TILs. Now, I think the question is, how do the NK cells even know that the tumor microenvironment is something significant? And I don't know if I have the answer to that. So in the final part of my talk, I just want to focus on using our assay to design trials. And so the one trial that we've done has been to focus on patients who are in this part of the spectrum, people who have low amounts of these partially exhausted TILs. Can we do some type of intervention and turn these cold tumors into hot tumors? And so I've been focused for many years on this molecule interleukin-12 because I feel it's a critical driver of Th1 responses, and I feel like that's the one way you have to attract T cells into tumors. And so we've developed this technology. Essentially involves injecting a plasmid directly into tumors and then using electroporation. And this is the kind of, essentially you cause small breaks to appear in the membrane and you drive the plasmid in. And so this is my colleague, Rich Heller, who developed this. And this is a photo many, many years ago. And so in our phase 1 trial, we showed that in 2008, that if you took a patient with metastatic melanoma, and if you injected 4 lesions in his case, this is a guy who had hundreds of tumors. So he had tumors on his front and on his back. And so if you did electroporation with interleukin-12, in this one patient, about 2 years later, he had complete resolution of all the tumors. And you could see that even on his back, where we didn't touch, he had this vitiligo kind of thing develop. And so we've replicated this in a phase 2 trial. And here are results from the phase 2 trial. So just showing that both in lesions that were treated and lesions that were untreated, you had some degree of response to single agent interleukin-12 electroporation, maybe about 20-ish%. But if you look at the progression-free survival, it's disappointing. It's not what you would expect to see, even though you have some degree of response. When we looked at these patients, what we realized is that many of these patients, although they failed the interleukin-12, they went on to get PD-1 treatment, and a surprising number of them, 6 out of 8 of them who had post-failure PD-1 had responses. And when we looked at their tumors, we realized that you were getting T-cell infiltration, even though ultimately the lesions were progressing. And so we asked Merck if we could combine it with pembrolizumab. And Scott is right here. He was instrumental in this decision. So this was I want to say in 2015. And the idea was to combine interleukin-12 electroporation with pembrolizumab. And this is the schedule. And patients were selected based on their being poor in these exhausted T cells. So they all had T cells at a level where we would guess they wouldn't respond to treatment. And you could look at the progression-free survival. The response rate was pretty high. It was like 43% by RECIST, and I think about 38% of those were complete responses, like you can see here. And this just shows their PE-TIL prior to treatment and the clinical response by RECIST or by clinical assessment. And I just picked this patient because I think it was an interesting patient to show. This is a patient who actually progressed. You could see that this is the gentleman here. And he had a bunch of tumors on his left leg. And he's a patient who had actually progressed on IPI, progressed on Nivo, progressed on Pembro after that. That wasn't me who did that, but he was when he came to us, he already had multiple checkpoint inhibitors. And so that's what we see a lot of times, that you could see that there seemed to be a progression initially, and then ultimately that patient responded. And my colleague, Alan Al-Ghazi, biopsied a whole bunch of these residual tumors and essentially just found some siderotic scars. And so when we look back at patients who were treated with either monotherapy interleukin-12 or the combination, essentially what we see is that if you look at the monotherapy patients had only a modest increase in CD8 infiltration versus a more significant infiltration in the combination therapy. But the real difference was that, again, PD-L1 expression was increased in the combination, but the real difference was right here in that last bar here. When you look at proliferating PD-1 positive T cells, and we see a significant increase in these, and this is in blood. So these are basically peripheral circulating T cells. And we think that what was happening in the monotherapy treatment was that you weren't getting a systemic response because of the development of adaptive resistance. When you block PD-1, you're getting more of those T cells proliferating in the periphery. And then this just shows the gene expression profile. And you could just see that if you look at Th1 or Th2 genes, you could see that there's just much more of a magnitude of change with combination therapy, both in the non-responders and the responders, but really overall you could see this difference. And this shows the overall, like the different gene expression profiles. And I'm just going to wrap up in a second, but so I see George looking here. But basically, we see a pretty profound T cell infiltration, both in responders and in patients who have progressive disease. The difference seems to be that is in the CD8 T cell to macrophage ratio. So you could see that on the top is a person who had progressive disease, and the bottom is a person who had a complete response. And you could see that there was actually a T cell infiltration in both in the treated and in the non-treated non-electroporated lesion. And the difference, just like you can see from the colors, you're getting much more of a macrophage infiltration in the non-responders, and those are the ratios. You could see in the responder, you're just getting a lot more T cells per macrophage, per M2 macrophage. And we think that this IRF1 to PDL1 ratio is going to be really important, because I think that has something to do with the capability of the tumor to respond to immunotherapy. And so we are working on what the implication is for treatment. So I think I'd like to conclude by saying these agents are incredibly active in cancer. That's a fact. They are great for combination therapies, but it's a complex issue, and even markers that seem to work for one checkpoint don't seem to work for other checkpoints. And I want to acknowledge both Mike Rosenblum, that's his lab here, and then our group up here, and my collaborators at UCSF. And thank you all for your attention.